Looks like most people are here now. So hopefully you're sketching this question or writing out this pathway. I'm going to give you another couple seconds and then we're going to go over it. So when we're talking about the electrical activity of the heart, we're just talking about basically this action potential that travels from our pacemaker cells through our conduction fibers. We have two nodes that we're going through. So the signal starts in our SA node, our sinoatrial node in the right atrium. The signals spread out through the atria, but when we're thinking about the pathway, specifically, they're going through the internodal fibers to the AV node. The AV node is at basically the bottom of the right atrium, right before the ventricles. AV stands for atrioventricular. From the atrioventricular node, we then go into the atrioventricular bundle, which is the same thing as the bundle of His. So that's starting to go between our two ventricles in the wall, called the atrioventricular septum. This then splits into two branches, the right and left bundle branches, going to our right and left ventricles. They go all the way down to the apex of the heart, the pointy bit at the bottom, before they give off the Purkinje fibers, which wrap up and around. The electrical signal travels in this order in a normally functioning heart. Normally functioning heart, that SA node is our pacemaker. So it beats the fastest. So despite the fact that these other fibers or these other cells could make a signal, the sinoatrial node wins out. So if we were to track that through the heart itself, I'm not going to step through slide by slide here like we did last time, but just take a quick look at the picture. We have the sinoatrial node up here. We have internodal pathways, so between the two nodes. They get us to our AV node, our atrioventricular node, which then gives off our AV bundle or bundle of His, starting to go between the ventricles, splitting into our right and left bundle branches, and giving us our Purkinje fibers wrapping around. Just so we remember some of the structure of the heart here that we can see. Remember, aside from the fact that we should know that the SA node is in the right atrium, we can recognize that this is the right side of the heart by the fact that we have this vena cava, this venous blood flow, blue, coming into the right atrium. You can distinguish it from the left atrium because the from the left ventricle, the left side, let's say. The aorta is coming from the left ventricle, kind of passes over the atrium here, arches around. We can see the ventricles that they're attached to. You'll note that the left ventricle is also slightly more muscular than the right ventricle. These are some things that you'll see in lab in the spring. Okay. So what we didn't get to last time was talking about how you actually can measure the electrical activity in the heart. So this is done with an electrocardiogram. So that's either an ECG or an EKG. The EKG is just when they've written it in German, cardio starts with a K in German. So those are equivalent. So an ECG can be recorded from electrodes on the skin. So we can actually sense this movement of electricity, these electrical pulses, these depolarizations, by placing electrodes on the skin itself. So they travel to the surface. This is great because we can measure heart function without actually getting in there, right? This is a non-invasive technique that can let us test for clinical abnormalities. So we've talked a little bit about how, right, the AV node would take over if the SA node fails. In order to figure out it failed, we might need to actually take a look at that electrical signaling. Okay. So this is an external measure of the electrical activity of the heart. And the body is conducting those depolarizations to the surface. So this is our ECG, is the heart one, but we've seen this in PhysLab as well as an EMG. So the EMG measured, right, electricity in our muscles, in our skeletal muscles, so we could see contractions of skeletal muscle. And we also have EEGs. We have all sorts of ways to measure electrical activity in different parts of the body. So the distance and amplitude of the spread, so what this graph looks like, depends on a couple factors. So one thing is it's going to depend on the size of the membrane potentials, so the size of these changes. It's also going to depend on how synchronized the cells are, because note that this isn't measuring like one cell in your sinoatrial node. This is going to be kind of an average from the collection of cells, right? We are just placing an electrode on the outside of the body. We can't probe a single cell. Okay, so which cells are doing things together makes a difference. Fortunately, heart electrical activity is synchronized. It does flow in this pathways. We have gap junctions between our cells. So this communication between our electrical pathways and our contractile cells, so the ones actually contracting, it is synchronized. So it gives us something that we can actually distinguish pretty well. Okay, so we looked before at the membrane potential of our contractile cells, right? So what's happening here? So that's what this graph on the top is. Here is how it lines up with what you would see in an electrocardiogram. Okay. So you've probably seen this familiar sort of series of little spikes on TV shows, movies. Each of these peaks and dips is given a letter so that we can talk about the distance between them, because each of these peaks and dips is going to correspond to an event, an electrical event in the heart. So the first thing we have happen here is this little P wave. So this P wave is the depolarization of the atria. So the atrial depolarization, so that's that top part of the heart. So as they receive this signal, right, that's spreading from the SA node, they depolarize. So we see a slight little peak here in our electrocardiogram there. Then we have what is called the QRS complex. So we have a dip, then a big peak, and then another dip. So what's happening during this QRS complex is that the ventricles are depolarizing, so that signal is getting to the ventricles, and the atria are repolarizing. So we see this big peak here, right? This big depolarization as our ventricles are depolarizing, and the atrial repolarization is in here as well. The T wave here at the end is happening as the ventricles repolarize, and we can measure distances between different parts of this wave, basically to look at how quick different events are happening. So we might be interested in, is the signal going really slowly between our SA node and our AV node, right? In which case we would look at the PQ segment. So the PQ segment is, because P is our atrial depolarization, and the QRS complex is our ventricles depolarizing as the atria repolarize. So if we were to look at this PQ part, that would be the time as that signal is passing to the ventricles. So that tells us about that pathway. The QT segment, so QT, This is basically the whole time that the ventricles are squeezing, right? Because this QRS is ventricular depolarization. So depolarization is the action potential, which is telling those contractile cells to contract, right? So if we measure from the start of that to the end, the T, the repolarization, that tells us how long the heart is squeezing. And that's really important because it's important to not tire out your heart, right? Your heart can get exhausted. So if we were to really elongate this period of systole, that could present problems for the heart. And then since Q to T is systole, systole is when the ventricles are squeezing. Diastole is when the ventricles are relaxing. I like to remember that because like when they dilate your pupils, right? Well, I guess maybe this only helps for people who have to go to the eye doctor, but when they put eye drops in and your pupils dilate, right, they get bigger, they get relaxed. So that's what that happens during diastole as well. Okay, so that TQ segment, so from the end of one of these little images, one of these little pulses to the next one, that's diastole, while things are relaxed. You can see that there aren't any really changes here in the electrical activity, right? As we're relaxing, we're not sending an electrical signal, we're not contracting, we're not having action potentials moving through these cells. So that's why that's this flat part, basically. So quick question about this. I'll share the poll question that goes along with this, just to make sure we're all awake before we move on to the next bit. Oh, I think, oh, sorry, I may not have actually put this on here. Where'd it go? Okay, I guess I didn't create a poll question for this. Okay, but what's happening during the QRS complex, right? Go directly back, QRS complex, ventricular depolarization, and atrial repolarization, right? So that would be C. Depolarization means, right, basically systole, we're contracting, we've sent the electrical signal to those cells in the ventricles. Okay. So I know this has been rapid fire after Thanksgiving, or at least it feels rapid fire for me. I don't know if I've fully recovered from break, but hopefully to this point we have a decent grasp on the order of blood flow through the heart and the order of this electrical signal. Now we're going to be talking about the pressures here. So this is kind of adding on to both of these things. So as we talk about this, we'll be reviewing a little bit the order of blood flow, but we're going to dig a little deeper. We're going to be thinking about why. So I brought myself a prop today. So I just want to make sure that we have a grasp on how pressure works, because I know some people are a little startled when we start I don't know, having equations or doing mathematical comparisons. But conceptually, when we're comparing pressures, it should be really easy. So I have a balloon here. Right, I'm going to blow it up. So I had to work kind of hard there. I guess my lungs aren't that strong. I had to put pressure in. I had to push to blow up this balloon. So we now have pressure in the balloon. There's pressure stored in the balloon. It's higher pressure than my environment here, than the air around me in my room. Okay, so when I release my fingers here, we all know what's going to happen, right? The air is going to come out. So what's happening here with the pressure is that the air is traveling from an area of high pressure inside the balloon to an area of low pressure, which is this outside environment. So I'm going to release. right? So it went from high pressure to low pressure. There's a little bit of air still in my balloon, right? So now there's equal pressure inside my balloon and outside my balloon. Yeah. So if you start to get tripped up a little or get a little nervous as we're talking about the pressures in the heart, that's the one thing you want to come back to, right? We're going from high pressure to low pressure. We're going to have different events happen as these pressures shift. So the other thing I could do with this balloon, right? Because we're going to be talking about the heart, right? If I squeeze on my balloon, I'm going to be increasing the pressure, right? I squeeze on it. So if I squeeze on it, as I'm letting the air out, right, we could even see that, right, because the air would come out even faster. So we're just thinking about pressures, and we're also going to be thinking about volumes and how that relates to pressure. Okay. So the cardiac cycle is going to be happening in phases. What we're tracking during these phases is what the pressure is, where that's coming from. We're going to be thinking about the relationship between the pressure and the volume. So another sort of conceptual thing that I'm going to demonstrate here with the balloon is that relationship between pressure and volume. So to blow up my balloon to here or so, that's not hard. I didn't have to really push very hard. It's when I try to expand this area, when I increase the volume in this same balloon, right? The balloon, the physical like rubber, I still have that same physical space. When I try to push more air into it, more volume, that increases the pressure, right? It got harder to push air in there as I tried to push more air, more volume in. So that's another sort of conceptual thing that we want to be thinking about as we're going through this heart stuff. As you put more volume, that's going to increase pressure unless we increase the actual space that's being filled. Okay. So the cardiac cycle, the overview for this, and the outline is posted now. Sorry about that, guys. We're going to go through the phases of the cardiac cycle. We're going to go through the pressure in the atria, but really we're going to be focusing on the pressure in the ventricles. The other place that pressure is going to be really important, and we're going to talk about it really specifically, is in the aorta. So we're going to have some neat properties of the aorta that allow it to basically maintain blood flow through the whole body, even while the heart is basically paused. And that's going to have to do with the pressure in the aorta. We're also thinking about the volume of the ventricles. Eventually, you notice when I release my balloon, right, there were sounds. We're gonna look at the heart sounds as well. Okay. So when I say cardiac cycle, we're talking about the events that are happening during one whole heartbeat. So one whole cycle is going to be like lub-dub, lub-dub. Sorry, those are my heart sounds. But between those two things, right, that's our one heartbeat. There will be two sounds during our heartbeat before we get to the next one. So that's going to be our cardiac cycle. When we're talking about the cardiac cycle, because there are a lot of things going on, the way we are going to define the phases really is dependent upon what's happening in the ventricles. So we're going to be thinking a lot about the ventricles. Because of this, we have, oh good, I am recording, I got nervous all of a sudden. Okay, so because of this, we're going to talk about contraction of the ventricles and relaxation of the ventricles as our two main parts of the cardiac cycle. We're going to see there are 4 phases, but like 2 main periods. Systole, is when we're contracting the ventricles. So systole, I don't know, like stress, you're clenching up. Diastole, ventricles are relaxing, they're dilating, they're getting bigger, they're relaxing. And fortunately, these are good concepts, important concepts to know, because these are also going to correspond to roughly some of the things we measure when we're measuring blood pressure. We'll see how that links up with pressure in the arteries as well. Okay, so our valves, which we took a look at in our lecture on the heart anatomy, our valves are really opening and closing due to pressure gradients. So I mentioned the AV valves, the right one and the left one, they do have muscles attached to them, but the muscles are not causing them to open and close. They're just to prevent them from prolapsing. What opens and closes the valve is purely about the pressure gradient. Okay. So the AV valves are going to open. So that's I think I'm going to do a fair bit of drawing today. So I'm going to draw my little stereotypical heart. So I got the atria, the ventricles. Here we'll have my other ventricle, other atria, right? Something like that. Okay, so here's my heart. So the AV valves, which are located between the atria and ventricles, all right? We have our tricuspid valve and our bicuspid valve here. These are going to open when the atrial pressure is higher than the ventricular pressure. So they're little leaflets. And when we have high pressure here, low pressure here, we just talked about, right? Things want to flow from high pressure to low pressure. So those valves open in this situation. So blood is able to flow when there's higher pressure in the atria, lower pressure in the ventricles. This pushes on those leaflets of the valve and allows blood to flow through. So AV valves are open when there's higher pressure in the atria. Our semilunar valves, and I'll draw on, I like the aorta. So this is coming from our left ventricle. Okay, so we'll have a semilunar valve here. Okay. Just like the AV valves, they're going to open and close dependent upon pressure. So when they're going to open is when we have high pressure in the ventricle. So it's going to go to an area of low pressure. It's not going to go back to the atria, right? Because the reason for those valves between the atria and ventricles was to stop the backflow of blood from the ventricle to the atria. So the place we're actually comparing the pressure to is in the arteries leaving the ventricles. So when we have low pressure in the arteries leaving, in this case the aorta, we're going to have high to low flow. So our blood is going to flow from high to low this way. This flattens down those leaflets against the wall of the aorta or the pulmonary trunk, the pulmonary arteries, depending whether we're talking about the left or right ventricle, but we're going to be kind of biased towards the left ventricle, to be honest with you. Okay, so when we have high to low ventricle to artery, that's when our semilunar valves are going to open. So I want you to be thinking carefully about these pressures as we move through the phases of the cardiac cycle. Always moving areas of high pressure to low pressure. And when that high to low is in the right direction for the blood to flow, that's what opens valves. If, I'm gonna just undo a couple, okay. So if we had the reverse of pressures, right? So if at some point we end up with high pressure in this part of the aorta, low pressure in our ventricle, we'd have the blood wanting to move back towards the heart, back into the left ventricle, because it just wants to go high to low, right? That's the only thing that matters for this blood flow. But we wouldn't be able to do that because that would slam our semilunar valve shut. So whenever the blood is trying to backflow to where it's just been, that's when we're going to see our valves closing. So we're going to talk about this in 4 phases as we go through the cardiac cycle. Okay. So I like to draw as we do this. So I will probably do a fair bit of drawing as we talk. So I'm going to draw myself a little heart. The pictures are on here. But you know, so I find it helpful for me to draw, for you guys to draw, even though we're not great artists necessarily. Okay. So we're going to start our number of phases with ventricular filling. So when we're talking about these phases of the cardiac cycle, really what we care about is the ventricles. And the reason for that is that the left ventricle is what's sending the blood out to the systemic circuit, out to all our organs. So that's really where we're super concerned about blood. So ventricular filling is occurring while the ventricles are relaxed. So they're relaxed. There's not much pressure in there. They're relaxed. So what we have happening is that blood is flowing from the atria to the ventricles because of pretty low pressure here in the ventricles. In the atria, the reason, right, that we'd have relatively higher pressure, although not very high in the atria, is that those atria are receiving blood from the rest of the body, right? So we have our vena cava, superior and inferior, bringing blood continuously to the atria. As there's more blood, as there's more volume, that increases pressure. Okay? So more volume in the same amount of space means more pressure. So as blood comes into the atria, raises pressure in the atria, which means blood flows into the ventricle. And this is passive. Our AV valve opens. This valve is open as the blood pushes on it, allowing blood to move into the ventricle. Note here, our pulmonary and our aortic valves are closed. Okay, so during this part of our cycle, I draw our aorta because I've just been drawing on the right side. Nope. Can't erase just that part. Okay. So lost my pen. There we go. Okay. So these valves are closed. Because they're closed, we're going to start raising pressure in the ventricles as they fill with a little more blood. Okay. Undo these lines, and we're going to move to our next phase. Okay. Okay. So our next phase is something called isovolumetric ventricular contraction. It's a mouthful, but we can break it down, right? So we have iso, which we've seen before, like when we were talking about osmosis. Iso means same, equal. So just like isotonic meant we had equilibrium on either side of a membrane. So isovolumetric means same volume. So this is telling us that we're having the same volume, but our ventricles are contracting. So what it's talking about when it says same volume is it's telling us that we have the same volume of blood in this ventricle the whole time. So we have no blood entering or exiting the ventricle during isovolumetric ventricular contraction. So that's the isovolumetric part. Is we have no blood. Our valves are actually all closed, right? I'll draw X's where we have a closed valve, right? We have a closed valve between the atria and ventricles. I guess I want my aorta on here again. Okay. And then we have our closed valves in our arteries. Okay. So we have the same amount of blood staying in those locations, but we do have ventricular contraction, right? So our ventricles are squeezing that heart muscle, those muscle fibers that are contracting. This is the start of systole. Systole is ventricular contraction. What's happening as those ventricles contract is we're increasing the pressure, right? So just like when I squeeze on it, right? Like I can squeeze a little, it gets harder to squeeze the more pressure I try to put here. As this ventricle contracts, we're increasing pressure because we're not changing the amount of volume, but we're squeezing harder and harder. So that same volume is getting kind of concentrated. Right. This is going to lead us into our next phase. Just clear all. It's simpler, which is ventricular ejection. So ventricular ejection, we're still going to have our ventricles contracting, but now we're going to have blood actually moving. So I'm going to draw my heart again. I actually mainly care about the left side here because that's where I'm going to draw my aorta. Okay, so here's my aorta coming out of the left ventricle. This is my interventricular septum. And here's my left atrium, just so you know what I'm drawing. Okay, so ventricular ejection happens because we have raised pressure in the ventricle by squeezing, right? We've created a system where we have high pressure here because we squeezed on that blood, compacted it in. And we have relatively low blood pressure in the arteries. So this allows our blood to flow, opening those semilunar valves that were closed before. So during our previous stage, our isovolumetric ventricular contraction, we didn't have this high to low gradient. We still had more pressure in the arteries. But once we've built up enough pressure in the ventricles here, those semilunar valves open. Okay, so now we have actual movement. So that's why it's called ejection, right? We're actually ejecting blood from the ventricles, from the heart. At the end of this phase, we're going to see that the semilunar valves close. That's going to happen when we equalize this pressure, right? As volume escapes, right? As blood leaves the heart, as it's being ejected, that lowers and lowers and lowers the pressure in the ventricle until eventually the valve closes. So eventually we would see equal pressure and then even lower pressure. So as these two equalize, as these two become equal, and then eventually the ventricle becomes low pressure, we'd have backflow of blood. It's going to get caught in these pockets, which is going to slam the semilunar valve closed. Our final phase of the cardiac cycle is isovolumetric ventricular relaxation. Okay, so iso, again, same, volumetric, same volume. When we say same volume, what we're talking about is the blood. There's no blood entering or exiting the ventricle. So we have the same volume of blood, but the ventricles are relaxing. So this is the beginning of diastole. We saw that the cardiac cycle started halfway through diastole, and now it's ending with the beginning of diastole. What's happening here is that, maybe I'll just draw the left side of the heart. Okay. Cool. So here's our heart, right? What we have happening is that as our ventricle relaxes, if I draw it now, as it relaxes, this volume, sorry, try to use the word volume just for the blood here so we don't get confused. The size of this space in the ventricle gets bigger. The volume of blood inside it stays the same. That's the isovolumetric. So this has the result of lowering pressure. Okay. During this portion, the AV valves and the semilunar valves are closed. And part of how we can know that from the name is that it's isovolumetric, which means no blood is flowing in or out. If those valves were open, we'd have blood moving somewhere. Those valves are closed because of the pressures, but also that's kind of what defines this, is that we don't have blood moving in or out. Okay, I think this poll I did put up. Okay, so quick check. What is happening during isovolumetric contraction? Okay, take a last second for this. Okay, so most people got this right, but I can see there's a fair bit of confusion here. So yes, the AV and semilunar valves are closed. That's what keeps this isovolumetric. Blood is staying in the same place. We're keeping the volume the same. The part that is contracting are the ventricles. So the answer here is D. When we're talking about the cardiac cycle, when we're naming these phases, we're really tracking what's happening in the ventricles. We refer to the other parts of the heart and the cardiovascular system when that's relevant for what's going on with the ventricular pressure, but we're focused on contraction of the ventricles. So we have this diagram, and I believe this is from your book. And this is linking up all the different things together. So this diagram is quite involved, but I'm just going to point out a couple of things. We're going to step through different parts of it. But what we're looking at here, right, this is showing us the phases, right? So the numbered phases, 1, 2, 3, 4. We have their names: ventricular filling, isovolumetric contraction, ventricular ejection, isovolumetric relaxation. We have arrows for where the blood is flowing, or I should say where the pressure gradient is, because during our isovolumetric ones, we don't actually have blood flowing. This is showing us the pressure pushing back towards the atria, but it can't go because the valves are closed. It shows us the state of the valves here in this portion. So it tells us the AV valves are open during ventricular filling. They close for the rest of the cardiac cycle. Our aortic and pulmonary valves, those are semilunar valves, are initially closed. Then they open because they're in the arteries during ventricular ejection. So that's when we're actually letting blood leave into those arteries, and then they close again. And then we have some pressure graphs that we're going to get into next. But this is telling us about the atrial pressure in purple, the ventricular pressure in green. We're also going to be discussing the aortic pressure, so the pressure in the artery, the aorta, here. So when I was writing those H's and L's for high and low, what I was comparing was the pressures. So this is telling you, right? So this is telling you that we have, yeah, it's showing us the relationships between the pressure. We'll step through it. Here we see ventricular volume, heart sounds we'll get to at the end of next time, probably. And here's our electrocardiogram at the bottom. So we can see how that links up with these phases as well. Okay. So when we're thinking right now, we're going to be comparing to start with the atrial pressure and the ventricular pressure during these phases. We're going to be comparing atria and ventricles, and then we're going to be comparing arteries and ventricles. So remembering our atria are on top, atria, ventricles on the bottom, and they got walls between them. Okay. So during our first phase, the atrial pressure is rising slowly as we fill with blood. The ventricular pressure is low. So we can see that there's not a huge difference here. The green is our ventricular pressure. You can see that the green line is slightly below the red line here at the beginning during phase 1. Blood moves towards the area of low pressure, so it moves towards the ventricles. So that's our ventricular filling. At the end of phase 1, so at the end of this ventricular filling phase, the actual final part is that our atria contract, right? So our SA node sends a signal to those internodal pathways, and the atria are contracting. So as the atria contract, all right, so as they get smaller, this makes the atrial pressure even higher. And we can see that little bump, right? So that little bump pushes that last bit of blood into the ventricles. So that's phase 1. Okay. So then during phase 2, remember that's our isovolumetric contraction. So isovolumetric, we're keeping that same volume of blood in the ventricle, but it's contracting, so it's getting smaller, right? So as it gets smaller because it's contracting, we're compressing that same volume of blood into a smaller area, which raises the pressure, right? So we can see that on the graph. So phase 2 is quite short, right? Here's phase 2, but we can see that the ventricular pressure is shooting up. The atrial pressure is going down a little bit, especially in comparison to the ventricles. This means that blood would want to flow from high to low, from the ventricle to the atria, but as it tries to flow backwards, this snaps our AV valves shut, so it can't. Okay. So then we got phase 3. During phase 3, our ventricular pressure initially goes up. So this is our ventricular ejection. So this starts as soon as the pressure in the ventricles is higher than the pressure in the arteries. So they're still squeezing for the first part of it, which is why we start to see, or sorry, they are still squeezing. They're squeezing on a lot of blood at the beginning, but then as there's less and less blood to squeeze out, the pressure falls. So that's what we see in this arch, right? They have a lot of blood that they're squeezing on here, and then the blood volume is falling because they've been ejecting it. The atrial pressure is continuing to fall. We don't, at this point, we don't care so much about the atrial pressure during this phase. What's going to be important here is the arterial pressure. Okay. Oh. All right. There's not a section for phase 4 here. So I'll just mention what's happening here during phase 4. You can see during phase 4, which is the isovolumetric relaxation. So that beginning of diastole, as our ventricle expands, as it will get even bigger, right? It's expanding, it's dilating. This drops the pressure super low, super fast. Right? So that's what's going on here. Okay, so we will pick up here next time talking about the aortic pressure. So we were just comparing the atrial and ventricular pressure. Next time we're going to compare that to the arteries that are leaving our heart.